Again, thanks a lot for the introduction. I'd like to thank also Dr. Zhang for giving me the opportunity to be here again. And I'm very happy to see that this Congress is getting every year more successful. So I think the idea of my talk is to explain the project that we are currently working out. This project aims to explore the safety and the effectiveness of the maternal spindle transfer technique. You all know this technique has been proposed to prevent the transmission of mitochondrial diseases and has been already applied clinically also by the group of Dr. Zhang. But we think it can also play an important role in the treatment of infertility problems related to poor oocyte quality. So as you all know, assisted reproductive technologies have evolved and can now circumvent many subfertile disorders. However, the efficiency is still limited by the number and the quality of these oocytes. Women affected by different dysfunctions at ovarian level or with severe endometriosis, advanced maternal age, we all know that are often challenging patients using IVF conventional treatments. And frequently, some of these patients present poor embryo development that persists even if the cycle is repeated several times. So discarding that the problem is related to a male factor, sometimes if this problem of oocyte development persists or embryo development arrest persists, this is attributed to a problem of the oocyte quality. At the moment, we don't have a good biomarker to predict if an oocyte is good quality or not. So this is often determined after insemination and judging whether the oocyte is able to make a good quality embryo, euploid, and with chances to implant and to sustain a pregnancy. This developmental potential of the oocyte is mainly dictated by nuclear and cytoplasmic factors. And in particular, the cytoplasmic machinery is composed by several organelles, by mRNAs, ribosomes, proteins, and many factors, some of them that are still unknown. The mitochondria are the most numerous organelle present in the oocyte and are thought to play an important role because if they are in such a big amount, they should play an important function. And actually, in a human mature metaphase 2 oocyte, it has been described that it can contain over 100,000 mitochondria or even several thousand copies of mitochondrial DNA. So there are several publications that have been tried to correlate mitochondrial dysfunctions with the oocyte developmental potential. Actually, the Van Blair showed that higher ATP levels seem to be correlated with a better embryo development and better implantation rates. Also, other studies showed the other side, that decreased ATP synthesis can also present adverse effects on spindle formation, on the fertilization, chromosome segregation, and resulting in poor embryo development. And also other studies have tried to correlate mitochondrial dysfunction or altered mitochondrial DNA copy numbers with female age, and this could explain somehow why aneuploidy rates and implantation failure is more frequent in this type of patients. So we know also that the mitochondria are normally inherited through the cytoplasm of the oocyte, and that mutations or deletions in the mitochondrial DNA can cause severe diseases. And therefore, it's important that it's important that the mitochondria that are present in the oocyte at the moment of fertilization are both healthy and functional. Healthy to prevent the transmission of mitochondrial diseases, and functional to ensure that the oocyte has enough energy to support the embryo development during the first early stages of the development. Because we know that since the oocyte is fertilized until the embryo implants, it's supposed that there is no replication of the mitochondrial DNA. So, knowing that mitochondrial dysfunction, as I just referred, seems to be a major cause for decline in oocyte quality, there were strategies that have been proposed in order to enhance the quality of the oocytes in patients with previous IVF failed attempts. So the first experiments were proposed by the group of Dr. Jacques Cohen by the end of the 90s, and these experiments were based on co-transferring a small amount of cytoplasm taken from a donor oocyte and injecting it together with a sperm into the patient's oocyte during ICSI. These experiments resulted in live pairs, but because they were doing heterologous cytoplasmic transfer, the result was that the babies were born with high heteroplasmy levels of around 15%, and therefore, because there was concerns related to these heteroplasmy levels, FDA decided to suspend or to ban this procedure. We now know that some of these babies that were born were followed up, and there's this report published in 2016 where showed that apparently these babies that are now around 20 years old don't seem to have problems related to the technique. Nevertheless, there are other groups that have been tried to still enhance the quality of those sites using mitochondria. This was another technique that was proposed using autologous mitochondrial transfer to prevent heteroplasmy. This technique, other authors even offers this technique commercially under the name that Augment. And this technique was mainly based on isolating cells that were presumably present in the ovary, called the ogonial stem cells, and they were isolating these mitochondria and injecting them in the oocytes of the patients with the previous IVF attempts. Failed attempts. So this was a way of preventing heteroplasmy levels. However, the results did not show significant improvements in the quality of the embryos, and actually a randomized pilot trial published by EV Group did not show any improvement at all. So, knowing all this, it seems that mitochondrial transfer per se is not sufficient to correct these dysfunctional mitochondria or to repair other cytoplasmic deficiencies that may be still present in these poor quality oocytes. So if we really want to enhance the quality or get rid of these poor or these deficiencies that are present in the oocyte, we might need to replace the entire cytoplasm. And this can be made by transferring the nuclear genome of an oocyte from this poor quality oocyte into a healthier cytoplasm. So to do this, we can use different developmental stages of the oocyte. We can use germinal vesicle nuclei. We can use polar bodies. We can use meiotic spindles or even pronuclei. And there are several publications that demonstrated in animal models and even in human oocytes donated for research that any of these techniques can work in vitro. And in our views, maternal spindle transfer is the one that probably has the greatest potential. This technique is based on using metaphase II oocytes. And we start by removing the meiotic spindle from the donor oocyte, and then transferring the spindle from the patient oocyte into the new cytoplast. Afterwards, the oocyte would be ready to perform ICSI, and if the embryo develops in a good quality oocyte, it could be biopsied and eventually transferred, and eventually it could result in a baby that would be genetically related to the patient. It's a procedure that requires good micromanipulation skills because it's technically very demanding. However, if all the steps are performed carefully, it's associated with low mitochondrial carryover levels. Apart, it offers the advantage that manipulations are performed in the oocyte before fertilization, so this is considered a positive aspect from an ethical point of view in some countries. And because we are using metaphase 2 arrested oocytes, it's easier to coordinate the spindle donor oocyte with the recipient cytoplasm. Usually there are also different ways of explaining this technique in a more relaxed way. So this is with cocktails called pisco sour. And first we nucleate and then we transfer this orange part into the new cytoplasm. So it's a more relaxed way of explaining the same. The real technique was first approved or authorized in the UK in 2016, and the HFEA, after performing a long evaluation about the safety and effectiveness of the technique, decided to approve to license a clinic in Newcastle. However, as far as I know, there are still no reports about pregnancies or live births from this group in Newcastle. Other countries are also discussing the possibility of regulating this type of technologies, like here in the US, Australia, Singapore, or Canada. And for the moment, I think the only baby that has been born using a maternal spindle transfer to prevent the transmission of a mitochondrial disease was the baby that was that resulted from the study from the Tozang in 2016. It was a baby, a boy that was born to prevent the transmission of a Leigh syndrome. In our group, we thought that part of this important application, spindle transfer, could also play a role in the treatment of infertility problems. So we started to explore this hypothesis some years ago with a proof of concept in the mouse model, where we showed first with sibling oocytes that we were able to transfer the meiotic spindle without impairing embryo development after insemination of the manipulated oocytes. We have also showed that in a mouse strain with a very poor reproductive outcome, that by performing spindle transfer we could overcome very significantly the massive embryo development arrest characteristic of this strain. Also, we've shown that these embryos that were created through spindle transfer were able to develop in vivo with a high efficiency, and that the heteroplasmy levels in the mice were around 2 to 3%, which was quite good, attending the size of the spindle in this species. Also, we showed that these mice that were generated in these experiments were able to grow perfectly and showed a perfect health. They were all fertile, and that the heteroplasmy levels in adult organs were stable. And after 3 generations, we were not able to detect any heteroplasmy at all. Knowing that the experiments were doing well, we then decided to collaborate with a group in Greece to do the translational project into humans. And this project started with a preclinical validation with human donor oocytes. And in this part of the study of the project, we showed within human donor oocytes that we were able also, after optimizing carefully all the steps of the protocol, that we were able to enucleate and to reconstruct the oocytes very successfully without impairing embryo development in terms of fertilization, blastocyst formation, or euploidy rates. Also, that we were able to do it with a minimal amount of cytoplasm when transferring the spindle from one oocyte to another. with carryover levels lower than 1% in about 30 embryos that were analyzed, and that we could do it using either vitrified or fresh oocytes, but the better results were achieved when the cytoplasm was used fresh. After that, we decided to go on and to start a pilot trial. All these experiments involving human oocytes, preclinical validation, the pilot trial, were authorized by the Greek Authority of Assisted Reproduction. We obtained approval consent from, consent from all donors and patients participating in the study. We also approved, got the approval from the ARB from the maternity hospital. All experiments were performed in Athens at the Institute of Life, and we delivered progress report updates every 3 months to the authorities to maintain them informed about the status of our project. Also, all patients that were involved in the pilot trial were not charged for anything of the treatment. So I guess you will want to see how is this made in real. So first is the nucleation or isolating of the spindle. This is performed in a microscope with polarized light that allows us to see the refringence of the spindle. Then the reconstruction is done creating a slit with a laser and leaving the karyoplast in the perivitline space. And after that, we apply a fusion protocol. In this case, we used the one that we tried that gave us better results, was the one described by the group of Dr. Mitalipov. So we decided to register the pilot trial, and the pilot trial had this inclusion criteria was addressed to women under 40 years old, and if you want, you can later on discuss why we decided to include this criteria. It was also, has inclusion criteria, it was for patients that had previous IVF failed attempts that could be correlated to impaired embryo development due to poor oocyte quality. And it has exclusion criteria. We put women over 40 years old and couples diagnosed with severe male factor. Also, we intended to recruit 25 patients. So the first case that we did was in a patient 32 years old. This was a low responder with a severe endometriosis. She had been operated 2 times and had 4 previous IVF attempts without embryo development at all. So we decided to collect oocytes and vitrify them in different cycles. And from these 3 oocytes, we warmed them, and we got 2 oocytes that survived to the warming procedure. The 2 oocytes had spindle, and we performed spindle transfer correctly. And then they were fertilized, and we got 1 blastocyst. This blastocyst resulted to be chromosomally normal and had less than 1% mitochondrial carryover. Then we decided to transfer this embryo, and resulted in a pregnancy. And at 16 weeks, we decided to do amniocentesis to confirm that the result of the chromosomal analysis was correct, which it was. And then we analyzed the amniotic fluid, and we compared also with the embryo in terms of mitochondrial haplotype, which confirmed to be similar in the amniotic fluid in the embryo and in the saliva of the donor, and different from the patient. And we also did forensic DNA fingerprints to confirm that the nuclear genome was correlated to the mother and to the father in different SNPs. The pregnancy resulted in finished after 39 weeks and resulted to be a boy. with almost 3 kilos and 51 centimeters. He was born in April, and now it has about 5 months. Every 3 months we are doing a follow-up and doing neurological exams and measuring the pteroplasmy levels over time. These results will be included in this registry that we have created specifically to this project, and that aims to allow us to keep a track on all babies that will result from this pilot trial. And we are also open to include information or to register the babies that can come from other groups working in this type of technologies. We have done also another complete case in another patient. In this case, was a patient with 39 years old with unexplained infertility, but she had 11 previous IVF failed attempts. We used 10 metaphase to vitrified oocytes, 8 of them survived. We were able to do correctly spindle transfer in 6 of them. 4 fertilized and obtained 2 blastocysts. 1 of them was chromosomally normal. We transferred it, and it also resulted in a pregnancy that is now about to deliver. It's expected for the end of this month. Another interesting case is this one that I'm presenting here. It's a patient with a polycystic ovary syndrome. She is 35 years old and had 5 previous IVF attempts. So we collected the 16 oocytes, and this case is interesting because she had so many oocytes that we decided to split them randomly for spindle transfer and for the control group. So in 9 metaphase, in 9 spindle transfer oocytes, we obtained 4 blastocysts, good quality, that were biopsied, and 2 resulted in euploid embryos. And in the control group, it was performed by the same operator and under the same culture conditions. We couldn't get any blastocyst at all. So this, one of these euploid embryos was transferred and also resulted in a pregnancy that is still early, but it's going well. So up to the moment, we have done 20 patients with an average age of 37 years old and average of previous IVF failed attempts of 4.9. And I think results are quite promising because the mean number of oocytes used was 4.3 per patient, and the successful spindle transfer rate was over 90% with a fertilization rate similar to that can be obtained in normal non-manipulated oocytes of good quality. We obtained good blastocyst formation rates and 45% euploidy rates. So we transferred also another patient, but the embryo did not implant. But I think for the moment, the numbers are still low, but the results are very promising, especially because we're dealing with patients that have a long history of previous IVF failed attempts. Of course, I'm going to present these results in more detail next year.
No, sorry. We have 2 more minutes.
I'm finishing, sorry. So I'm going to present these results next week at the SRM. Of course, this is still an experimental procedure, and usually the introduction of new technologies in our field are sometimes controversial. We have been following all that has been suggested by in this publication by the Special Interest Group of Ethics and Law and Security in ART from ESRE. And here they suggested to use a tool to score and to understand the experimental phase of when a technique is introduced in humans. So for the moment, we have done all these steps where we showed the safety in the mice. The results are about to be published in high-impact journal. We have showed in preclinical with human donor oocytes that the fact of transferring the spindle from one side to another is not affecting the further development of the oocytes. We are doing this in a very strict restrict cohort of patients. Of course, we obtained all the consents from the authorities, and we have obtained consent also from the patients to follow up the babies that will come from this pilot trial. So just to conclude, I think it's clear the spindle transfer is feasible and does not adversely affect the spindle apparatus or early embryo development, but of course all the steps of the protocol are a bit tricky. They require a lot of practice and need to be optimized carefully. Spindle transfer can enhance the potential of the developmentally compromised oocytes without compromising euploidy rates, and this can be done with very low heteroplasmy levels. Our results in the pilot trial are preliminary, but seem to indicate that the spindle transfer oocytes are able to implant and to stay in a pregnancy to term. And also we need to go on and do more carefully controlled pilot trials to provide more insights into the feasibility of the technique and for clinical applications to prevent the transmission of mitochondrial diseases or to overcome infertility. Also, if everything proves to be efficient and safe, I think this technique can open new hopes to patients with infertility problems that cannot be treated with current clinical strategies, offering them the chance of having a baby genetically related to them, and could also be advantages for donors because the resultant children would not be genetically related to them, so it could reduce their psychological concerns that they have at the moment, and could reduce the concerns related to the anonymity. That is an issue that has been discussed now in Europe and could also provide some benefits in this regard. So I'd like to thank everyone involved in this project, and also you all for your attention, especially because this is the last session and you might be tired already. So thank you very much.